Hello and welcome to the Lymphoma Research Foundation's Understanding CAR T-Cell Therapy and Lymphoma webinar. My name is Kathy and I'll be your operator for today's call. During today's call, you will hear from expert speakers. You will have an opportunity to ask questions. If you have any questions during today's presentation, you can ask them at any time in the Q&A box on the webinar. As a reminder, this webinar is being recorded. At the end of the program, a link will appear on your screen. Please follow the link to complete the evaluation of this program and gain certificate of attendance. If you are listening by phone, this link will be sent to your email at the end of the webinar. I am now pleased to introduce Jesse Brown. Jesse Brown is the Associate Director of Patient Education at the Lymphoma Research Foundation. Welcome, Jesse. It is all yours.
Great. Thank you so much. Thank you, and thank you to each of you for taking the time to join us on today's Understanding CAR T-Cell Therapy and Lymphoma webinar. I'd like to thank our sponsors of this webinar, Bristol Myers Squibb, Kite, a Gilead company, and Novartis Oncology. Before I turn the program over to our speakers, I want to briefly share information with you on the Lymphoma Research Foundation. Access to expert disease information is so important, and we're thrilled to be able to bring you this educational program. LRF is the nation's largest nonprofit dedicated exclusively to lymphoma. Our mission is to eradicate this disease through investment in the most promising lymphoma research and to serve those impacted by lymphoma through quality education and support opportunities. As we continue to make progress in advancing lymphoma research, we also want to ensure that you have access to the latest information about your disease. The Foundation provides comprehensive disease and treatment-specific resources, programs, and services, all of which are offered free of charge and have been reviewed by lymphoma experts. Most relevant to today's call, LRF offers a variety of lymphoma-specific resources, many of which you can access at the bottom of your screen if you're utilizing the web link, or via LRF's website at lymphoma.org if you're on the phone. The LRF helpline can answer your specific questions about lymphoma, as well as discuss relevant treatment options and clinical trials. We also offer the Lymphoma Support Network, which is a one-to-one peer support program for people with lymphoma and their caregivers. We also offer a variety of publications that have been reviewed by lymphoma experts to ensure you have access to the latest lymphoma information. We have comprehensive books on understanding NHL, HL, CLL, and the transplantation process. Our mobile app, Focus on Lymphoma, is an award-winning app that provides patients and caregivers access to comprehensive content, as well as unique tools to help manage your disease. Finally, we've launched our COVID-19 Learning Center to support lymphoma patients and caregivers this challenging time. Please visit our Learning Center for access to webinars, articles, and other resources specific to COVID-19. I really hope you'll take advantage of some of the great resources and services that LRF provides. If you have questions regarding what you heard today, or if you need information about relevant treatment options and supportive care resources, you can reach out to the LRF through our website at lymphoma.org or by calling our helpline at 1-800-500-9976. We have a wonderful program planned for you today, and I'm honored to introduce you to Dr. Javier Munoz. Dr. Munoz is a hematologist and oncologist, as well as the director of the lymphoma program at Mayo Clinic, Arizona. He's been the principal investigator in many CAR T-cell therapy trials at Mayo Clinic and has been an expert speaker on many LRF patient programs in the past. Thank you so much for speaking at our program today, Dr. Munoz. I'll now turn the talk over to you.
Good afternoon. Thank you for inviting me. Thank you for your attendance. My work gravitates around lymphoma, immunotherapies, and CAR T-cells. Happy to share my experience, and thank you again for joining us. I have the title of the presentation right there. What is in store for you? We have divided the presentation. I have three topics. Number one, understanding CAR T-cells. Number two, when to consider CAR T-cells for patients with lymphoma. And finally, managing potential side effects. This is the first part of my presentation. The immune system is the body's defense against infection and cancer. This immune system is made up of cells that are divided into several subtypes. Lymphocytes, a subtype of white blood cell, they come in several flavors. For example, you have B lymphocytes or B cells that make antibodies to fight infections. We also have T lymphocytes or T cells that have several functions, including helping B lymphocytes, but T cells also directly kill infected cells or abnormal cells in your bodies. Throughout the presentation, I'm going to have these small graphics that represent the good guys and the bad guys, represented here as a police officer and a thief. But in general, the CAR T cells in this particular graphic are on the left, those T cells that you have re-educated to seek and destroy the malignant cells. And the malignant cells on this graphic is on the right. The CAR T cells that are currently approved are targeting B-cell lymphomas, which means that the lymphocyte that went originally rogue and started duplicating more often than it should, it was a B-cell lymphocyte. Despite the simplicity of the graphic, this is a very sophisticated process and extremely exciting development in medicine. You are weaponizing the T-cell lymphocyte to attack a B-cell malignant lymphocyte. So it is a sibling attacking another sibling. How to make it easier to process or understand this new therapy? Let's try an animation. How do CAR T cells work? Again, remember the graphic on the left side, you're going to have the good guys. That's why you have the police officer, in this case the T cell. On the right side, you are going to have a tumor cell. You are going to modify genetically the DNA of that police officer, of that T cell. You are going to re-educate it in the way that you're going to teach that cell how to go after a particular marker. In this case, we have chosen CD19. Those are the CAR T cells that are currently available in the market, but there are many clinical trials looking into other targets and other diseases. But once you have re-educated that T cell, you're going to express this construct in the surface of the cell, and it's almost like a Velcro that is going to facilitate that the good cell is going to get attached or is going to interact with the bad cell, with the malignant cell in this case. The CAR T cells now are able to recognize that particular tumor cell antigen. As I said, in this particular example is CD19. You need to be careful when it comes to which target you choose because you want the market to be primarily located in malignant or abnormal cells. You wouldn't want the market to be widely expressed in normal tissue because you could launch an autoimmune attack in a way after infusing the CAR T cells. These CAR T cells, after making contact, they're going to multiply and replicate. That's why we call it a living drug in a way. And subsequently, they're going to produce these cytokines, these interleukins, these substances that we all produce, let's say, when we get a common cold. And ultimately, this interaction is going to make those tumor cells to die, something that we call apoptosis, cell death. So in a nutshell, that is the process that I wanted to show. Now, CAR T cell, CAR is an acronym, C-A-R, which means that every letter stands for a word. So CAR means chimeric antigen receptor. Sounds like a mouthful, but I'm going to try to dissect what it means. Chimera, let's start with the first word. It comes from mythology, Greek mythology. It's a fire-breathing she-monster described by Homer, and it had three parts. It had part of it was a lion, it was a goat, and it was a snake. So those are the three parts from the chimera from Greek mythology. So let me show you the three parts of this CAR T cell. There's an external domain. Again, you need to choose it wisely. Multiple clinical trials looking at multiple targets in liquid tumors and solid tumors. But as I said, the ones approved at this point in lymphoma would be anti-CD19. So they are looking for that particular target that is expressed on the surface of the cancer cells. You are going to have an internal domain that would be stimulatory. That means that it's going to make, after that CAR T cell makes contact, you're going to activate those T cells to make them duplicate and replicate and so on and so forth. And then there's a hinge. There's a third part of the chimera. They are co-stimulatory domains as well. So these constructs are expressed on the surface of the T cells, and they are going to recognize a marker that is expressed on the malignant cells, that in this case is CD19. Think of it as a Velcro, putting those two cells together. All right, second part of of my presentation would be when to consider CAR T cells for lymphoma. And I can see some questions coming up about different types of lymphomas and leukemias. So I'm hoping that this is going to help some of the attendees. So who is going to benefit from it at this point is based on the label, is based on the FDA approval, which comes from the inclusion and exclusion criteria of the pivotal trials that leads to those approvals. So again, there are multiple diseases under investigation, but there are only very few situations where we actually at this point we have the approvals, and this is a crescendo of development. And I'll show them here in a second. The first CAR T cell FDA approved for lymphoma was Axi-cel. But actually, the first CAR T cell approved period was Tisagen for B-cell acute lymphoblastic leukemia. So they both were approved in 2017. Subsequently, in 2018, Tisagen, and of course, I'm shortening the names, the full scientific names of these CAR T cells because they do not really roll off your tongue. I mean, they're very complex, but I'm shortening them as Axi-cel and Tisagen. Tisagen was the first CAR T cell approved period, initially approved in 2017 for B-cell acute lymphoblastic leukemia. The first CAR T cell approved for lymphomas was Axi-cel, same year, 2017. Around the same time, we had the approval of tocilizumab. Remember in the initial animation that I showed you, once those two cells make initial contact, the immune system revs up. It gets activated, so you start producing all of these substances, cytokines and interleukins. So they noticed that one of the interleukins that was elevated was interleukin-6, and there was a medication in the market that is an anti-interleukin-6. So as an antidote, when the immune system revs up too much, something that we call cytokine release syndrome, we also have a medication FDA approved to counteract those effects, which is an anti-interleukin-6. So this is the initial snapshot of that journey for CAR T cells that started in 2017. And this is what we had even as of mid-2020, last year, when I gathered this slide, we had only those two, but things have changed since then. But we had the Axi-cel and the Tisagen, and if you look at the minutia, you will see that the label was not exactly the same. Why? Because again, the clinical trial, the inclusion-exclusion criteria was different. I have highlighted the difference for you there. Primary mediastinal B-cell lymphoma is within the label of Axi-cel for relapsed refractory patients. It is not present for Tisagen. For Tisagen, you also have relapsed refractory acute lymphoblastic leukemia. As I said, it is not present on the label for Axi-cel. Many other diseases, even though they may have the word lymphoma within their names, if they were not part of that initial pivotal, of those initial pivotal studies, they would not be part of the indication at that point. If you look, for example, at the fine print at the bottom, it clearly says, for example, it does not include primary central nervous system lymphoma, which means that this is not a blank check in a way that is not, the label is not for all lymphomas. It is only for the lymphomas that were studied in those initial trials. So let me show you some additional granularity. Recently, with that I mean last month, February 5th, there was a third CAR T cell FDA approved for aggressive B-cell lymphomas. And you could, if you go to the fine print of what the label states, there are some differences when it comes to Tisagen, Axi-cel, and Lyso-cel. Primarily when it comes to Lyso-cel, you also see follicular lymphoma grade 3B, and also you see that transformation from indolent lymphomas to aggressive lymphomas is also part of the verbiage when it comes to that FDA approval. Now, there is a fourth approved CAR T cells last year, July 2020. We got the first CAR T cell FDA approved for patients with mantle cell lymphoma, relapsed refractory. That wording is important too. I say this because many of you, and we have had this, I mean, CAR T cells are sexy, they're in the news, people talk about it, there's a lot of noise about it. So after the approvals, even in 2017, we had a lot of patients call us with different types of lymphomas or leukemias asking for CAR T cells. But again, if the particular subtype is not present on the label, then this is not a right treatment for your particular disease. And also the setting, it needs to be relapsed and refractory. We had patients that would call us saying, I had diffuse large B-cell lymphoma 10 years ago, I got R-CHOP chemotherapy, I have been in remission for 10 years, can you give me CAR T cells? And the answer is no. You need to have relapsed refractory disease. That means that the disease needs to be coming back. You cannot get it if you are in remission. And also you need to have the right disease per label, which again is based on the inclusion-exclusion criteria of the pivotal trials. So this is a cheat sheet. It's a little bit of a summary that will help you perhaps identify the different types of lymphomas that are out there. Again, the disclaimer is that there are more than 60 subtypes of lymphomas. This is a very humble summary, just highlighting the ones that are mainly approved at this point. But if you have a rare disease, like I'm reading in the chat, LGL leukemia, that one is not at this point approved. I appreciate the questions for sure, but again, if you have a disease that is not part of this list, it wouldn't qualify. And also, if you are under observation and or in remission at this point, that wouldn't be part of the label as well. So I hope that this table is helpful. On the lower part, you see the crescendo of development when it comes to the years when the approvals occurred, and this is helpful for us, the doctors, to remind ourselves when the CAR T cells are a good option for our patients. And this is the last part of my presentation. I only have 3 more minutes to go, so I'll try to be brief. Managing potential side effects. Again, what is the toxicity? It's not a walk in the park, so again, you never want to ask for something that you do not need. We said it's a living drug, so you have these T cells, these police officers duplicating, replicating, and I gave you the analogy of perhaps when you have a common cold, the immune system activates and you have these T cells produce cytokines and interleukins. How do you feel when you have a common cold? You're fatigued, you're achy, you're tired, you just want to sleep in the couch. Well, multiply that times 20 or times 30, and that's what can happen after CAR T cells. Sometimes, not always, but it mimics sepsis. You look like you're infected even though you may not be infected. So on the lower area, and actually I believe I have a little graphic here, this would be a representation of the cytokine storm. And as I said, among the many interleukins and cytokines that get elevated or increased within your peripheral blood, you could find interleukin 6, and that is why you have this anti-interleukin 6 in the market called tocilizumab. And on the lower area, you will be able to see a graphic that explains how this cytokine storm, this hurricane, as you can see in the graphic with the cytokines, is not immediate. Usually it takes a couple of days for these cytokines to be increased and cause symptoms that mimic infection, as I said, fever, low blood pressure, low oxygenation. And once those symptoms start to get better, sometimes patients get neurologic events. Why do we get neurologic events? We do not understand it very well because we know that there's no CD19 expression, let's say, in the brain. So it's probably somehow associated to interleukins and cytokines as well. And I know in the next presentation, Dr. Bevan is going to show you more granularity, but again, it's not a walk in the park. It's not for the faint of heart. These are the numbers for one of the constructs, for axi-cel. severe cytokine storm, 13%, severe neurologic events, 31%. Different constructs are going to have different degrees or numbers when it comes to toxicity, but the bottom line is that it is not a walk in the park necessarily. And how do we manage this toxicity? What do we do? This is just to show you. I'm not going to read line by line, but the bottom line is that there's a method to our madness. We spell it out. We exactly know what to do if the oxygen starts to decrease or if the blood pressure starts to become a little crummy. We know exactly how to act. It's usually a combination of that antidote that I told you, that anti-interleukin-6 that would calm down the storm, so we can ride the storm in a way. And also steroids. Steroids are lymphocytotoxic. That means that they destroy lymphocytes. And if you have a lot of T lymphocytes duplicating, replicating, steroids would be a poor man's way of destroying those lymphocytes and bringing them down when it comes to their numbers. So hopefully the storm can ease down as well. And this is the bottom line, the conclusions. Again, immunotherapy has a unique response pattern. It also has a unique toxicity pattern. Multiple clinical trials in development. There are downsides. Again, there's the cytokine storm, there are neurologic events, and also cost is an issue. The long-term toxicity is unclear. We don't have decades of experience in some scenarios, but I do think that the future is bright for patients with lymphoma. I'm happy. It has been an honor to present the basics of CAR T cells to you. Dr. Bevan's presentation will provide more granularity and sophistication when it comes to this topic, and she'll go a little bit deeper when it comes to the details. I'm happy to take questions, if any, at the end of the presentation. Thanks again for inviting me.
Great. Thank you so much, Dr. Munoz. That was wonderful. I'm now honored to introduce you Dr. Ann Bevan. Dr. Bevan is a hematologist and oncologist and lymphoma program director at the University of North Carolina Lineberger Comprehensive Cancer Center. Her clinical research interests include Hodgkin, non-Hodgkin, and T-cell lymphomas, and she's heavily involved in the development of the CAR T-cell therapy program at UNC Health. Thank you so much for speaking at our program today, Dr. Bevan. I'll now turn the talk over to you.
Great. I wanted to thank everyone for coming today. So the first thing you're going to see in this talk is that part of the reason I went to med school is I have absolutely no art talent, and you'll see that when you see how beautiful Dr. Munoz's slides looked and how plain mine looks. But hopefully it will still have lots of information for you. A lot of my talk is actually going to be talking about the future of CAR T, so I'm going to talk a little bit about some things that are investigational, and I'll try to really point those out. So my understanding in talking about this was that a lot of what I was going to talk about toxicity-wise was really what are the kind of long-term toxicities. So Dr. Munoz has already talked about what can happen in the sort of 2 to 4 weeks after CAR T. And so I wanted to talk a little bit about what happens further down the road. So in general, one thing that is really exciting for CAR T, and not everybody on this talk has gotten chemotherapy in the past, but for those of you who have, you know that when you are getting CAR T therapy, when you're getting chemotherapy, you might get it for months at a time, sometimes even years at a time, where you're having to go in for the medicine, you're having to take pills, you have these long-term toxicities. One of the really wonderful things about CAR T therapy is it's sort of this one-time thing where you have to be followed very closely when you get the CAR T, so that you're being seen maybe multiple times a week. Sometimes you might be in the hospital for a few weeks, but it's really this sort of 4-week time when you're being monitored so closely. And after that, people do really well, toxicity-wise. Dr. Munoz mentioned the cytokine release syndrome, which can be severe, and I'm not going to talk about that very much now, other than to mention one thing, which is because of the tocilizumab drug that he mentioned multiple times that can help to decrease that cytokine release syndrome, I think that we are getting better and better at handling cytokine release syndrome. In the initial studies of CAR T therapy, they were concerned that if they did anything to stop that cytokine release syndrome, that kind of fighting from the immune system against the lymphoma, that it would make the CAR T less effective. And so they would let people get pretty sick with the cytokine release syndrome before they would intervene. And now in sort of clinical trials and clinical practice, there's expectation that you really respond to that and intervene much earlier. So we can hopefully keep many patients from getting really sick with the cytokine release syndrome. The other thing that I'd say with the neurotoxicity when it occurs, which is the thing I see most frequently in my practice, is people, if it happens at all, it tends to be people getting very sleepy, kind of confused. I see this more often in my older patients, as you might expect. But the neurotoxicity happily seems to be almost completely reversible in well over 95% of patients. So long-term toxicity, the good thing is that the long-term toxicity that we see with the CAR T therapy is actually very limited. So the two things that I see in my patients sort of long-term, once we get really out of that 4 to 6 week window, is one thing called prolonged cytopenias. So for those who've gone through chemotherapy, you'd probably be a little bit more familiar with this. So when I talk about cytopenias, I'm talking about low findings in the blood work, like somebody who's anemic, somebody whose infection-fighting cells are low, if your platelets are low, which are part of the blood that helps to stop bleeding. Often just after chemotherapy, we'll see these things get low. And as part of the CAR T therapy, patients will get 2 or 3 days of chemotherapy, which can cause the cells to get low. But what we see is that after CAR T therapy, sometimes the cells stay low longer than we would expect with just traditional chemotherapy. Part of this is probably because people who get CAR T therapy have been treated so many times that their bone marrow is maybe just a little bit more tired. It takes it a little bit more time to recover. But it seems that there's probably some effect from the CAR T cells as well. But the happy news is that even when patients have these counts being low, in my experience, clinically, it doesn't really mean that much to them. So this is kind of a small slide, but basically it's looking at results from 2 of the trials that have been done for FDA approved agents, so the Axicell and the Tisagen. And what they found was that at 28 days after getting CAR T, about a quarter of patients still have pretty low neutrophils, pretty low infection-fighting cells. And even out at 3 months, in one of the studies, 10% of patients still did. And then as far as platelets being low, out at 28 days, you have about a quarter to almost half of patients where those will still be low. But really, it sounds like once you're getting out to about 3 months, we're in most cases seeing improvement in these counts. In my experience, that people haven't had complications, all it means is that maybe they're having to go to their doctor more often to get their blood checked, and occasionally someone might need a transfusion. The other thing that we can see is suppression of normal B lymphocytes. So as Dr. Munoz explained to you, the CD19 target is on B cells. So that means it's on normal B cells, normal B cell lymphocytes, as well as on B cell lymphomas. And so what happens when you get this CAR T cell, this new kind of immune fighting cell, and it looks for CD19, it's binding to the lymphoma cells as well as the normal B lymphocytes. This is called on target, the target being CD19, off tumor effect. So it's doing the tumor, but also the normal B cells. So you can have results in a decreased number of B lymphocytes. The reality is that rituximab, which is a drug that so many patients with B-cell lymphomas have already gotten, also attacks the normal B lymphocytes. And so many patients, these B cells are low even before getting CAR T therapy. But the good news is that even if these are low, people do very well. So most of my patients who have low B cells or these low lymphocytes will have no problems at all. If they have problems, it is typically increased risk of kind of upper respiratory type infections, recurrent sinusitis or bronchitis. The other thing that, and if they have this, so if I have someone whose B cells are low and they're having recurrent infections, then those are patients occasionally I do have, once a month, give them either an IV or a shot medicine of these proteins that can help replace the function of those B cells. The other thing that patients are asking about quite a lot now is response to vaccinations. So these B cells, normal B cells, part of what they do is help to respond to vaccinations. So when they are low, this could decrease response to vaccines. It wouldn't make the vaccines more dangerous, but people are asking about it a lot, of course, because of the COVID vaccine these days. So that's something you would just need to discuss with your, treating physician. In the studies, the average time or the median time until the lymphocytes increase is about 9 months after CAR T therapy. So almost 2/3 of patients had more detectable B cells at 9 months and 3/4 at 12 months. So the rest of my talk is really going to be about future uses of CAR T therapy. We know it's FDA approved for mantle cell lymphoma, it's FDA approved for diffuse large B-cell lymphoma, but there's a lot of other lymphomas that are out there, some of which people have already been asking about in the question box. So what about some of the slow-growing B-cell non-Hodgkin's lymphoma, like a follicular lymphoma or a marginal zone lymphoma? Then there's Hodgkin's lymphoma. Then there's a completely different type of lymphoma called a T-cell lymphoma, which can be a peripheral T-cell lymphoma with inside lymph node involvement, or a cutaneous T-cell lymphoma with mostly skin involvement. So the axi-cell, several of these CAR T products have been studied in relapsed refractory indolent lymphoma. So this would be like a follicular lymphoma or a marginal zone lymphoma. These are still B-cell lymphomas, so they still express that CD19. So that's why we can use these same CAR Ts to try and target them. And what they found is that the responses are really exciting in these lymphomas as well. So this is just data from one of the studies where they have about 100 patients, generally older, so median age of 63, with the oldest patient treated with 79, which just tells you a little bit about tolerability. And they're pretty heavily pretreated, so on average, they'd had 3 prior lines of therapy. These are not people with newly diagnosed disease, and 3/4 of them didn't even respond to their most recent therapy. They were refractory to their most recent therapy. And the fact that patients were so heavily pretreated and still had good responses is what makes CAR T therapy so exciting. So what they found was an overall response rate of 94%, meaning that almost everybody had some benefit from the CAR T therapy, and 79% of patients had a complete response, meaning their PET scan improved from the left, where you can see lots of black spots, all representing lots of involved lymph nodes, to a normal PET scan on the right, where all of those lymph nodes are gone and the only little black areas are left are normal things like the kidneys and the bladder and the heart. So a complete response where we find no evidence of their lymphoma on their scans. So we've also looked at other CAR T products in follicular lymphoma with similar patient population, heavily pretreated, and again, demonstrating again very high response rates, 80% of patients responding and 65% of patients having a complete response. I added this because I think that one thing that is really interesting with CAR T therapy is that sometimes patients can have a response that's kind of slow. It's because it's your immune system working to fight this lymphoma. And so in this trial, in the follicular lymphoma patients, when they assessed for response 3 months after the CAR T therapy, some patients who were in a partial response, when they checked them again at 6 months after the CAR T therapy, had gone from just having some response to having a complete response at 6 months. So even patients who early on just have a little bit of response can continue to have improving responses. And I think that this is helpful. So what you're looking at here is along the bottom, you have time frame in months since the CAR T was infused. And then on the y-axis, you have the probability basically of what's called progression-free survival, has people's disease progressed. So you can see that I've put in this arrow here at the 6-month time point. And the reason I've done that is to the left of this arrow, you can see this sort of continuous decline. Every time that line drops down is a time when somebody's disease has relapsed. And what you see is that once you hit this 6-month line, this is for follicular lymphoma, that after you get out to 6 months, it's very rare to see relapses. And I think that this is something that we're consistently seeing with CAR T therapy and diffuse large B-cell lymphoma as well, which is that generally if people get to 3 months, if people get to 6 months, we're finding that those remissions are really durable. And so I just think that that's exciting news to share with people. So basically what we know is CAR T therapy in clinical trials seems to be very effective in indolent non-Hodgkin's lymphoma. It is not FDA approved yet, although the FDA is actually potentially looking at this later this week, so we may be hearing more about that in the coming month. And again, it would be for relapsed or refractory disease. So what about the future of CAR T? So next steps are looking at different targets other than CD19. So we can look for maybe at other types of cancers. Can we use allogeneic CAR T cells? So that means instead of what we do currently, which is where we have to take the patient and collect that patient's cells and go through a long process to make those cells, can we use just a normal healthy donor and make this process move more quickly? And other things are, what's the best timing for CAR T therapy? It's approved for when people have been relapsed multiple times, but in a high-risk patient, for example, people in the questions section have several of them asked about double hit or double expressing lymphomas. For those patients, should we use this as some sort of a consolidation earlier on? If somebody relapses, is this something we could consider as consolidation instead of a transplant? So those are questions that are still being investigated, and we're very early in the thinking about timing. But this is very early research data. But I just thought to give you guys a little bit of information about where we're going and other things we're looking about in CAR T. So CAR T has been studied in relapsed refractory lymphoma. So we at UNC, along with Baylor College of Medicine, have been looking at a CAR T that targets CD30. So Hodgkin cells don't have CD19, but they express CD30. And so that's why, in this case, that's the target we look for with the CAR T cells. So as you can see, this is a tiny number of patients, 42 patients treated with Hodgkin's lymphoma, so much more research is needed. But I just wanted to show you that there's hope for these other lymphomas as well. So 42 patients really heavily pretreated with a median of 7 prior treatments. 32 out of 42 had a prior autologous stem cell transplant. A quarter had a prior allogeneic stem cell transplant. These are patients who've really gone through a lot. And what we found is with this CAR T, it seemed very safe. We had very minimal cytokine release syndrome, 24%, and it was all the lowest grade. No neurotoxicity. The only thing we really saw was rash in these patients. But what we found was what we think were very high response rates. So 62% of patients had a response, and the complete response rate in half of patients. So these patients with an average of about 7 prior therapies, half of them going into complete response with this CAR T. And we haven't had years and years of follow-up, but when we last looked, the median progression-free survival, so the time where half of patients were still disease-free, was over a year. So there are, still Hodgkin's trials, studies of CAR T, larger trials going on to try and confirm, is this really something that we can offer in the future to our patients? CAR T is also being investigated in relapsed refractory T-cell lymphoma. Some T-cell lymphomas express CD30. So at UNC, we're looking at CD30 doing this, but T cells also express CD4, CD5. So people are looking at a lot of different CAR Ts in T-cell lymphomas. We don't have any sort of data for phase 2, phase 3 studies to really know what's going to be the safety, because it's probably going to be different, and what's going to be the efficacy. All we have so far is, a few case reports showing that there is at least some hope that we'll be able to find CAR Ts that may be effective in T-cell lymphomas. So on the bottom, you can see the PET scan with that bright white area. That's the patient's lymph nodes before infusion, which are cleared up after infusion for a peripheral T-cell lymphoma. And then on the right, you can see a patient, A and C is before and B and D is after, a patient who has skin involvement of their lymphoma, seeing clearance. So the other thing that people are looking at is whether we can use allogeneic CAR T cells. So I mentioned this briefly before, but right now, the way CAR T works is you take a patient who has, let's say, diffuse large B-cell lymphoma, and if you want to do CAR T, that patient has to generally go to a university setting and once they get insurance approval, they have to have some of their own cells taken away, drawn off through their IV, and those cells are what are sent off for the processing to make them into CAR T cells. So that can be a delay. For most of the FDA-approved indications, it's usually once the cells are collected, you can usually get them back to give back to the patient about 2 to 3 weeks later. If somebody's in a research trial, often those are can be much longer. And so this is very time-consuming, and you have patients who are ill, who have their lymphoma progressing and needing to wait around 3 weeks to get their treatment back. Sometimes it's kind of long. The other thing is that when you are collecting cells from a patient who's been treated multiple times with prior chemotherapies, sometimes their bone marrow is kind of in rough shape. Sometimes their T cells are sort of in rough shape. And so when we go to use that patient's cells to make the CAR T product for them, sometimes we can't make the CAR T product because their cells just won't grow the way we need them to. And the other thing, you can imagine, this is quite an expensive process, going through all of this for one patient over and over. So when we talk about allogeneic CAR T cells, what we're talking about is, can we, instead of using the patient's own cells, can we use sort of a normal healthy donor's cells? And so potential advantages of this, you can sort of see this little diagram that someone made where you have way over on the left the healthy donor, and you do the same process. You collect their cells, you send them off to the lab, you make them ready to target CD19 or whatever, and then you grow, duplicate all of those cells, so you can then separate them out into multiple bags available to give to multiple different patients sort of when they need them. So it would be sort of more of an off-the-shelf thing. The doctor just writes the prescription, and it's not quite this easy, but the product is ready rather than having to collect from each patient individually. So you wouldn't have those same delays. It could be potentially a little bit less expensive. So this is something that's very early in the investigational stage. And so we don't know, is it going to be more effective, less effective, and how will things be toxicity-wise? We worry a bit about increased toxicity because you have somebody else's immune cells going in to fight the lymphoma, but it's possible that these new immune cells will also look at the patient, their normal intestines, their skin, or wherever, and say, this isn't usual for me, and that this new immune system would target those also. So you may have increased toxicity. You can also have decreased efficacy. Maybe you get this other person's CAR T cells in your body, and your own T cells look and say, hey, I don't recognize these T cells, and your own normal cells attack the donor T cells. And if that happens, maybe they could be actually less effective CAR Ts. So that's really early in investigation, but something that people are looking at now. And so in summary, CAR T continues to be a very exciting and effective option for patients with B-cell non-Hodgkin lymphoma. Research is ongoing to develop CAR T therapy for Hodgkin lymphoma, T-cell lymphoma, and I should tell you, it's also multiple myeloma. People are looking in solid tumors, breast cancer, glioblastomas. A lot of research is going on to see how far we can expand the use of CAR T for cancers. And then we're hoping that eventually there may be an off-the-shelf CAR T option that could be a little easier for patients to use. And I guess that is it. So I think we can just go to questions and answers.
Yeah, thank you so much, Dr. Bevan. We'll now begin our Q&A portion of the program. Just as a reminder for everyone in the audience to please keep your questions as general as possible so that the entire audience can benefit from them. If you'd like to ask a question, please submit your question through the Q&A box on your screen. I can see a lot of you have been doing that throughout the presentation. And we'll take as many questions as possible, but if you have a question that does not get answered, you can always reach out to LRF helpline at 800-500-9976. And Dr. Munoz and Dr. Bevan, I'm going to alternate the questions between the both of you, but if either of you would like to chime in after the other one's question, if you have to add anything, please feel free. So our first question is for Dr. Munoz. This is from Joseph from Washington, D.C., who asks, how well is the cytokine release syndrome being managed now? Do patients still require ICU stay?
Thank you for that question. And I think it's a process. There's a learning curve. I think we're all getting more comfortable with CAR T cells. because it's not only the oncologist that is involved. I mean, sometimes you have hospitalists, internal medicine doctors, intensivists in the ICU, cardiologists, neurologists. It truly takes a village to take care of our patients, and I think all of us as a team were more comfortable taking care of our patients. But to answer your question, I think that at the beginning we were really afraid of, for example, using steroids, because you have these preciously manufactured CAR T cells, these lymphocytes that you have modified genetically, and if you would infuse them in the patient and you just too easily prescribe high-dose steroids, we were afraid that the steroids, again, they are lymphocytotoxic, so we would destroy these precious cells too quickly, too soon. So at the beginning of this journey, we were not prescribing as much treatment as we are now. There have been some retrospective studies. We look back at the patients that received steroids versus the patients that did not receive steroids, and we noticed that the outcomes are similar. So with that, we became more comfortable managing the toxicity, because we're no longer afraid that giving tocilizumab or steroids would somehow dampen or decrease our response rates. And patients still respond. You can control the storm as much as you can. If you remember what I quoted, it's not the majority of patients that develop severe cytokine storm, is the minority. I mean, one study says 13%, another study says 15%, another study says single digits. But the bottom line is that it's the minority of patients that would require or develop severe toxicity, which means that only a subgroup from those numbers that I have just quoted will eventually require intensive support in the ICU. And it's my experience that number has decreased as the years went by, with all of us getting comfortable with this treatment. Thank you for your question.
Great. Thank you, Dr. Munoz. Our next question is for Dr. Bevan from Joe from Virginia, and I know you talked about this a little bit already, but maybe you could just expand on it a little bit. He asked, what are the challenges in using CAR T-cell therapy to treat T-cell lymphomas?
Well, so one of the big challenges with CAR T therapy for T-cell lymphomas is that you need to figure out how to create a CAR T to target a T cell, like the T-cell lymphoma, that doesn't also target the T cell that's making up the CAR T. So there's a theoretical issue that if you just target T cells, will it impact the T cells that are the CAR T also? Will they kind of kill themselves? So that could affect efficacy. Certainly the other thing we worry about is if we target T cells and we suppress normal T cells as well, we worry much more about could we have more severe infections than we get with just lowering the B lymphocytes. So I think that there's some concerns with the toxicities also.
Great. Thank you so much for expanding on that. Dr. Munoz, Heather from Rhode Island asked, if responding well to CAR T-cell therapy, how long does one or should one expect to undergo treatment? Is the goal remission or cure specifically in mantle cell lymphoma?
That is a great question. I'm really hesitant in general to talk about the C word, cure. Of course, we all want that. That is one of our goals. But many of you have had that discussion, right? I mean, someone gets diagnosed with diffuse large B-cell lymphoma. It is a disease that we can potentially cure, so we all strive to achieve that even with R-CHOP. And many patients achieve complete remission, and many patients indeed are cured after frontline R-CHOP, but not everyone. So that is why I'm really hesitant to commit to that particular verbiage, because who decides? At the end of the day, time decides. If you received R-CHOP and 10, 15 years, 20 years go by and you never received another treatment beyond R-CHOP, then yes, you are among that subset of patients that likely got cured from their lymphomas with frontline chemotherapy. But the opposite is also true. If someone achieves a complete remission with R-CHOP and the first scan looks great, but months later the scans show that the disease is coming back, then it's a disservice almost to commit to the verbiage of cure. As I said, time is the best judge. As years go by, that you stay in complete remission without needing additional therapies, then yes, it is possible. I believe that it's possible that CAR T cells could potentially be curative for a subset of patients with whether it is these aggressive B-cell lymphomas like diffuse large B-cell lymphoma or mantle cell lymphoma like was mentioned in the question. But I would like to be careful not to say that this is one size fits all. There's probably some differences based on not only on the patient but also when it comes to the disease, maybe the molecular makeup of each disease. So I hope that answers the question.
Yeah, thank you so much. Dr. Bevan, a question from William, who's tuning in from New York, asks, who has the best response to CAR T-cell therapy? And as a follow-up, what options are available if CAR T does not work?
Well, I'm not entirely sure if that means who patient-wise or which drug. I'm not entirely sure about the question. But so the first thing I'd say is, as far as, is there one drug that's better than another? Because now we have multiple CAR T therapies that have been FDA approved, and these are all drugs that have not been compared one to the other, so we don't know if there's one that's better than any of them. I'd say that in general, overall, the toxicity of the different agents and the response rates seem fairly similar. So there's not sort of a belief right now that one is the superior agent. And then as far as, which patients might be most likely to respond, I think that, and when you look at the trials, sometimes they're seeing patients who are very refractory who respond. Sometimes patients who are newly treated respond. It's not clear in most kinds of drugs that we do for lymphoma. For relapsed disease, we tend to think people who have had longer remissions before, so whose disease didn't come back as quickly, or people who've had fewer lines of therapy are most likely to respond. That hasn't been as clearly shown in CAR T, that sometimes we are seeing people with highly refractory disease who respond to that as well. As far as what's available after CAR T therapy, a lot of that depends on what patients have gotten before. In the past year or two, we've had a couple of other agents that have been FDA approved for relapsed refractory diffuse large B-cell lymphomas. There are definitely other options that are out there that have been having pretty good efficacy. So there are other things. Another thing that is really investigational that's being looked at is, can we combine CAR T therapy with other regimens? I think I saw a question from somebody talking about PD-1 inhibitors like nivolumab or pembrolizumab. There's some data suggesting that maybe using those with CAR T or after CAR T could improve efficacy by improving the immune system. So I think that there's still a lot to be determined as far as, is there a best drug to be given if someone relapses after CAR T, and how to proceed afterwards.
Great, thank you, Dr. Bevan. Dr. Munoz Prasadi from Sydney, Australia asks, if the tumor expresses antigen, what could cause CAR T to not respond as expected?
Excellent question, and I appreciate the commitment joining all the way from Australia. I'll have to figure out the differences, but thank you so much for joining and for your excellent comment. So what we think is that there is something called antigen escape. So you are targeting CD19. If the CAR T cells would do a wonderful job obliterating or destroying all the cells that are CD19 positive, then in theory you could have a clone that is CD19 negative, it does not express that particular marker, and that clone starts replicating, duplicating to the point that eventually builds momentum, and you see in the scans progression of disease. So it is possible that if you destroy all the cancer cells that have CD19, maybe there's a clone that has that does not have CD19 but has CD22 or CD20, and that is why some people are developing tandem CAR T cells. I noticed there were some questions in the chat about that. So to counteract, at this point, all the 4 CAR T cells that we have talked about, they are attacking CD19, so one target. They are under investigation, not FDA approved at this point. People are looking at other targets and combinations of targets. So people are looking at CD19 plus CD20 or CD19 plus CD22 and many other permutations. So it is possible that by doing that you could circumvent, you could counteract that issue that perhaps there's a clone that is CD19 negative. But the short answer is that we do not know very well. There are patients that still relapse, and you perform biopsies after the CAR T cells, and you find that they do still have a little bit of CD19 expression. So both scenarios can occur. You could have a patient that relapses with CD19 positive disease, or a patient that relapses with CD19 negative disease. And it seems that the explanation is within the realm of that antigen escape that I was discussing. Thank you.
Great, thank you, Dr. Munoz. Dr. Bevan, Chia-Chun from Washington State asked, since we have 2 CAR T products for relapsed refractory lymphomas, is there any clinical trials looking at receiving one, such as Axicell, then the other, Lysocell, if relapsed later? How far apart can you receive another product?
So I would say at this time, I'm not aware of any clinical trial that's looking specifically at somebody getting one of those CAR Ts and then later getting the other. In general, I think that getting CAR T is probably something you do once. Now, in the future, what if we have people and we find that as time goes by, people get CAR T and 5 years later they relapse? Will we say, maybe it's useful to give someone CAR T again? I mean, maybe that's something we'll be looking into the future, or other things within one of the trials that we have going on right now for peripheral T-cell lymphoma at UNC. One thing that we're doing is, as part of the study, we're giving patients a CAR T. This is really investigational, and then we give them a second dose of the CAR T, a few months later, looking at, okay, can you increase efficacy by doing it that way? But a specific question of if someone gets one of the FDA-approved agents and then, should they get another one in the future is really something that we don't know the answer to yet.
Great, thank you. I think we have time for 2 more questions. Dr. Munoz, does Medicare cover CAR T-cell therapy? And if not, are there other ways to seek out treatment in a more affordable manner?
That is an excellent question. The short answer is yes. I mean, once you have an FDA-approved therapy, this should be allowed to be prescribed across the board. Disclaimer is that, of course, this is not, how can I say this? It's that these discussions are better had with your physician or your team face-to-face, because, of course, we do not know the minutiae of your insurance and or insurance situation. So it is true. I think that I understand where the question is coming from. It is tricky sometimes when it comes to the insurance situation. We as physicians, as prescribers, we definitely did not go into medicine to be the ones managing or addressing these issues. We do have a team, a financial team in each institution that they would look at the insurance situation. But the answer is yes. Once a product is FDA approved, it should be covered by the insurance. The fine print within your insurance details, that is something that goes outside of I believe the goals of this discussion.
Great, thank you, Dr. Munoz. And Dr. Bevan, our last question of the day is, what is the guidance on timelines for COVID-19 vaccine and CAR T-cell therapy?
So this is a great question, and I will tell you that there is debate all over the place about what should we tell our patients with lymphoma, our patients with cancer, our patients who are getting chemotherapy, because on the one hand, you know, I was listening to Dr. Fauci talk at our hematology conference last year, and he said if people are on immunosuppression, they should take COVID vaccine anyways. It's not more danger. Maybe it will have some benefit, even if less benefit than if they weren't on chemotherapy. But we know that B lymphocytes are involved in responding to vaccine. And so we know that those are decreased from CAR T therapy. We know they're decreased from rituximab. So with lymphoma in patients, B-cell lymphoma patients in particular, this is something we talk about a lot. And so what I found is that the recommendations are really all over the place. There's no consistent recommendations. So some places will say, I don't think you should get the vaccine for, 3 months after getting rituximab, or 3 months after getting CAR T or a transplant. Others will say 6 months. Some will say a year. Some say just get it now. Maybe it'll be some benefit is better than no benefit at all. So I think that that's really something that each person should talk to their particular doctor about to kind of get their input and recommendations.
Wonderful. Well, thank you both again so much, Dr. Munoz and Dr. Bevan, for joining us today and sharing your expertise on this topic. I know everyone on the call really appreciated you taking time out of your day to do this. So thank you again. And thank you to everyone for joining us on today's call as well. We hope you found the information both informative and also hopeful. We'd also like to thank our sponsors again for making this program possible: Bristol Myers Squibb, Kite Gilead Company, and Novartis Oncology. Please remember, if you have any additional questions or you'd like to be connected with someone else who has been impacted by lymphoma, you can reach out to our helpline at 800-500-9976. Also, at the conclusion of this program, you'll receive an email prompting you to complete a program evaluation. I'd ask that you please take a few moments to complete this, as they are very important for helping us to ensure that we deliver the most useful and meaningful programming to you. And with that, I'd like to thank you all again for joining us, and have a wonderful day.
Thank you.